Hi, let's talk about the action spectrum of photosynthesis. In 1880s, a scientist named Theodore did an interesting experiment. He was studying a filamentous algae under the microscope. Now, those old school microscopes usually had a mirror to reflect the light on the specimen. So this guy did an interesting experiment. The aim of this experiment was to find which colors of light were most efficient in carrying out photosynthesis. So he used a prism to get rainbow of colors. The 7 colors of rainbow were then allowed to fall on the specimen, which was placed on the stage of the microscope. Once this setup was complete, he then mixes a culture of motile aerobic bacteria on the slide. Since the culture is aerobic, it loves the presence of oxygen. And wherever the concentration of oxygen is more, these cells will migrate. After some time, he noticed the migration pattern of these cells around the filamentous algae. He discovered that the part of filamentous algae which was illuminated with red and blue color showed maximum clustering by the tiny aerobic cells. There were hardly any cells near the green region of the spectra. This experiment demonstrates that the red and blue color region of the spectrum shows maximum photosynthesis activity. So the action spectrum can be defined as the wavelength of light which is most efficiently used to carry out photosynthesis. Now, the action spectrum can also be represented in the form of graph. On the x-axis, we plot the wavelength of visible light. On the y-axis, we plot the rate of photosynthesis, which is usually the quantity of oxygen evolved. This graph has two distinct peaks, which corresponds to the maximum photosynthetic activity near the wavelength of blue and red part of the spectrum, whereas the wavelength corresponding to the green part of the spectrum shows minimum photosynthetic activity.